I will introduce your introduction, then we will start.
Yeah, that's great.
Good evening, everyone. This is Veer Maheshwar Charitable Trust Latur MS India, organizing daily international webinar series. This series covers the topic related to education, law, research, environment, health, technology. All topics are accepted here. And this series is running from last 5 years. We are inviting the speakers from India and abroad. All speakers are accepting our invitation and they are delivering their topic as per their interest and research of study. In today's session, we have invited as keynote speaker Mahtab Razlansari ma'am, who is Ph.D. in Organic Chemistry, MSc Nanoscience from Tübingen University. And her topic is Stimuli-Responsive Nanomaterials, the future of smart drug delivery. So this topic is related to chemistry and nanoparticles and our health also. So we welcome today's speaker, Mahtab madam, and she is a researcher with expertise in chemistry, nanoscience, and molecular biology, specializing in nanomaterials of drug delivery. She enjoys working at the interface of the discipline of tackle complex scientific challenges. She has invited from various organizations to deliver the topic. She has published the papers and she is an active academician and researcher. Madam, I welcome you from my organization and all the participants, those who are joined here. And we request you can start your speech now. Thank you.
Hello, everyone, and good afternoon. Thank you so much from Dr. BETKAR for inviting me to this international webinar series. Today, I'm here with an interesting topic about stimuli-responsive nanomaterials and the future of smart drug delivery. A brief introduction, as you said, my name is Mahtab Razlansari. I hold my Ph.D. from Rasi University in Inorganic chemistry. And also, I got my master in nanochemistry from Rasi University and my second master from Tübingen University in nanoscience. And thank you so much for joining this webinar. Okay. I organized this seminar for about 30 minutes, and for the 10 minutes of the last of this presentation, we have enough time for the question, and I'm very happy to answer your questions about this topic. And also, I ask you that please mute your microphones, and at the end, I will be happy to answer your questions. In this presentation, I will briefly introduce the concept of smart drug delivery and what is the stimuli-responsive systems, the internal triggers and external triggers. And we will talk about the different kind of nanomaterials, inorganic and organic carriers, and also the recent studies in this field. And at the end, we will talk about the challenges and limitation of these methods, and also the future prospects. And at the end, the conclusion and the question and answers. How stimuli-responsive systems works? Maybe it's your question, what is the nanocarriers and why we want to use them? Nanocarriers acts like a protective or physical platform for different kind of cargoes, for example, drugs, genes, imaging agents, and they protect these cargoes and transfer them to the target. And it depends on the treatment that we want to choose. For example, if we want to treat a cancer, we can have a variety of choices. For example, we can choose liposomes, we can use mesoperissilicos, MS nanoparticles, polymeric nanoparticles, and different kind of choices we have. And these stimuli-responsive systems are responsive to the special kind of triggers. And these triggers cause some changes to these materials. For example, they can make structural changes or bound cleavage and also gate opening. And the outcomes is very important for us because we want to deliver and transfer the drug and cargoes to the specific location in a specific time and manner. And also our aim is to increase the therapeutic effect and decrease the toxicity of this kind of materials. And the other question is, why do we need the smart drug delivery system? What is the problem with conventional drug delivery, like pills and injections that we use? The conventional drug delivery have some limitations. For example, they are not specific. They are non-specific biodistribution. And they have sometimes systematic toxicity. They are not very good soluble in the body, in the blood. And they will be very rapid and very soon will be cleared by the immune system. And also they have most of the times low therapeutic indexes. And it is the reason why we want to use the smart drug delivery system, because they have many advantages. They are responsive to the different kind of triggers, and we can control the release of drug. And specifically, we know that, for example, if someone have a brain cancer or any other kind of cancers, we know that the location, where is it? And this kind of nanocarriers deliver the drug exactly at this site. So they are very precise. And it causes that we have a minimal of target toxicity and also improve the therapeutic index. So they are very safe and non-toxic for the normal cells. There are different kinds of triggers for this nanocarriers. The endogenous stimuli-responsive system, they are internal. We don't need any external triggers for this kind of nanomaterials. They are internal. For example, the pH. pH can change something in these nanocarriers. They are responsive to the pH. For example, in our body, in the normal cells, the pH of the blood is 7.4. But in the cancerous cells, the pH is 5 to 6. The pH of the environment is acidic. So we can design and synthesize a specific kind of nanostructures that are sensitive and responsive to this pH. And as you can see, it's a picture, an image from a very interesting article. Here, it's a course structure, and it is designed with the polyethylene glycol ligands, and we have a TAT, and this are changing after encountering with the acidic pH. And as you can see, the ligand are changing the structure, and the releasing the drug will happen. At the other picture also is the same. The TAT are hiding inside the ligands, and after encountering in the acidic pH, these ligands will be released. And the structure TAT, which is good for penetrating the cells, are ready to act. And also they can reach the cells, entering the cells and releasing the drug. The other one is the redox responsive system. The GSH or cancerous cells, we have a higher concentration of the glutathione, and this increase in the cancerous cells. And when this kind of nanocarrier encounter with the cancerous cells environment, everything changed. The structure of them changed. For example, here we have a SS bounds. These bounds are be reduced by GSH, and they become the TO bounds, and the drug will be released. Okay, the next one is the enzyme responsive system. As you can see here, we have a bacteria. The active bacteria make some enzymes, lipase enzyme, And this kind of nanocarriers are responsive to these enzymes. And in counter with the lipase enzyme, they release the drug and the structure will collapse. So the bacteria will die easily. And what is the exogenous stimuli-responsive system? These kind of triggers are external. For example, we can synthesize a specific kind of nanocarriers which are responsive to the light, a specific wavelengths, for example, ultraviolet, near IR, or visible and different kind of lights. And these lights can break some bonds, cleavage the bonds, or change the physicochemical properties of the nanocarriers and change the structure. And after changing this structure, the drug will be released. And it is exactly what we need in these smart systems. The other one is the temperature responsive system, which is very interesting. We can design a special kind of nanocarriers. For example, here we have hydrogels. In the normal temperature, they are steady, firm, okay, they are stable. But when we increase the temperature, for example, 40 to 42 centigrade, they change their structure and they release the drug. This increasing the temperature is completely intentional, and researchers and doctors do it. And it's called hypothermia. The other one is the magnetic field responsive system. We can synthesize and create special kind of nanocarriers which are responsive to the magnetic field. For example, iron oxide are responsive to the magnetic field, and we can guide them. For example, if we have a core structure, the nanoparticles have a core of the, for example, iron oxide, they are responsive to the magnetic field, and we can guide them to the specific location. And the other one is ultrasound responsive system. Here, it's also very interesting. We can use ultrasound to create these bubbles, and this bubbles cavitation happens and releasing the drug to the specific site. So we have this kind of exogenous stimuli-responsive systems, and it is very useful because we can release the drug precisely and to the specific location, specific time, and also the intensity of the drug can be adjustable. Okay, now we want to know what is this stimuli-responsive nanocarriers. There are two most important groups, and we can categorize nanocarriers to two main groups: organic and inorganic systems. As you know, organic systems are based on carbon, and they have some similarities with our cells in our human body, for example. We can use different kind of nanocarriers as liposomes, polymeric nanoparticles, polymeric micelles, dendrimers, protein-based nanocarriers, and DNA or RNA nanostructures. They are very good and best choice for us if we want to do a gene delivery, or also for delivering the drug. For the gene delivery, they are very suitable because they are very biocompatible, biodegradable, and their structure is similar to the cell membrane, for example, lipids layer, and we can use them for different applications. The other group is inorganic nanocarrier. It's very vast, and you have lots of choices in accordance to your need. What kind of cancer, what kind of treatment do you need? You can design this kind of nanomaterials and nanocarriers. For example, mesoporous silicon nanoparticles, which have lots of pores, lots of surface area. You have a great capacity for loading the drug. It's a very good choice for loading the drug and delivering it and we can functionalize them also. The other one is the gold nanoparticles, which is very famous, and there are many researchers about the gold nanoparticles for treating different kind of cancers. The other one is the iron oxide nanoparticles. Magnetic nanoparticles, as I told you, they are responsive to the magnetic field, and we can guide them easily. So they are very famous and very used a lot by the scientists. Quantum dots, carbon nanotubes, graphene oxides, and calcium phosphate nanoparticles. For the carbon nanotubes, they have a very great structure. Tubes, you have a high capacity for loading the drugs, and also you have the structure. Carbons, you can attach the ligands on it and functionalize the surface. Ligands are very important in this topic, and I will talk about it in the next slide. And also calcium phosphate nanoparticles are also interesting. I read about them in some of the articles. They use it for bone cancers because they have a calcium phosphate elements, and they have a near structure to the bones. You can see the image of this kind of nanocarriers in this picture. They are very interesting and different shapes. Design strategies of stimuli-responsive nanoparticles, nanocarriers. As I told you, we can functionalize the surface of our nanocarriers exactly in accordance to our need. For example, on the cell membrane cell of the tumor cell, we have a different kind of receptors, and most of them are overexpressed. For example, we have a folate receptor on cancerous cells, and we can design a nanostructure, nanocarrier, which has a ligand and folic acid. This folic acid and the receptor folate acts like a key and a lock. They will be attached to each other and attaching to the surface of the cell, and they can get the allowance or permission to enter the cell. And it is one of the ways that we can help our nanocarrier to enter the cells. And after that, there are some process that happen, and they can release the drug at a specific site. And we can use some of the different linkers which are sensitive. For example, as I told you, they are pH responsive, ultraviolet light responsive, and different kind of linkers like disulfide bonds that I told you with the GSH, they reduce to the tile, and some structural changes happen and drug will be released. The other one is the gatekeeper system. They are sealed, the nanocarrier is sealed, and the drug is inside the structure. So when it is circulated in the bloodstream, they do not release it everywhere, the drug, but they will release it in a specific site. When they reach the specific location, these gates will be open, and the drug will be released at a specific site. And they are also sensitive to the different triggers. The other important things is about the biocompatibility and biodegradability of these nanomaterials. When these nanomaterials are done, I mean that they deliver the drug, everything is finished successfully, we need these nanocarriers to be cleared. We don't want them inside our body anymore. So they should be biodegradable, and they should be degraded in a controlled manner and very fast. So it is very important not to accumulate in the body for a long time, and it should be rapidly cleared. The application of the stimuli-responsive nanomaterials are very vast, and there are millions, thousands of articles and research about these nanomaterials. But the most important, and our focus in this presentation, is about the therapeutic effect. The cancer therapy, the gene delivery, which can be DNA, RNA, mRNA, the imaging and diagnostic, and combination therapy. It means that we can have a nanocarrier that have lots of properties at the same time, simultaneously. For example, we can see and we can treat. It is very important at the real time for the scientist to observe what is going on in the body of the patient that is going to be treated. And we call it trenostic. It means therapeutic and diagnostic at the same time. Here I selected some of the most important nanocarriers in cancer therapy. The first one, Doxil, is the first one exactly, which was invented by the scientist, and it got all the approved, and it was approved by the FDA and also EMA, which for the European Medicines Agency, and also Food and Drug Administration from the US. So it is used in the clinic. The drug is the doxorubicin, and our nanocarriers in this structure is a polyethylene glycolate liposome, and it is used for treating different kind of cancers, as you can see, ovarian cancer. The other one is the Abraxane. There are many kind of other approved nanomedicine that you can search it in Google easily and find it. But here I just listed some of the most important and selected ones. Abraxane used the nanocarriers, albumin nanoparticles. Albumin is the most abundant protein in the body, and the drug paclitaxel was used in this nanosystem. It also has got the FDA and EMA approved, and it was used for breast and lung and pancreatic cancers. And you can find the references at the last slide in my presentation. If you are interested to study more about this kind of nanocarriers which are used clinically, you can refer to them. There was a very interesting topic, and these nanocarriers had lots of advantages for us. And we were very eager to work on them. But always for every kind of materials in science, there are some challenges and limitations that we have to find a solution and tackle the problem or overcome these obstacles to get the final and ultimate product. The first one, synthesis of these nanomaterials, it's not easy. It's complex. The design of them, the synthesis, it needs a great knowledge, and also it needs a great fund. You have to get a support and budget to do these researches, and it's needs a long time also. The other one is the producibility. Sometimes they are not reproducible. It means that if you synthesize a specific kind of nanoparticles, for example, you synthesize the magnetic nanoparticles with a specific size, for example, 10 nanometer to 200 nanometer. At the first time, everything was okay. You did the characterization, and TM and SM results was okay. For example, 50 nanometer, the average range size of them. But at the second time, when you are trying to synthesize exactly the same procedure, exactly the same environment, but unfortunately you get the other results. For example, the size changed to the 60, 70, and it is the problem that many scientists faced it during the synthesis and designing of these materials inside the lab. So the other problem is the reproducibility, and also in a high scale and in a high yield. The other problem is that this kind of nanomaterial are not always safe. And we are talking about human body. it's very complex, and we cannot use and apply every kind of nanomaterials inside body. So, and our immune system is very sensitive, and will track them, and most of the time will clear them from the body and don't let them to release the drug or do their action. So they may have these problems as well. The other problems is about our understanding from in vivo behavior. For example, we synthesize something, but at the end, we predict that everything will be okay and the cancer will be treated. But at the end, we got another results because we are not sure and we don't know exactly what is going on in the body. But I suggest that before synthesize and designing this kind of nanomaterials in the lab, first of all, doing some stimulation and in the special softwares to know what is the result, to predict the result. There are many softwares that can help you in this way. The other one is getting the approved and regulatory and clinical translation hurdles. It means that there are many obstacles to translate a nanocarrier from the lab bench to the clinic. And if you want to invent something, you have to overcome all these obstacles. And what is the future perspective for this smart drug delivery? We are trying to develop the nanocarriers, which are stimuli-responsive nanocarriers. For example, they are responsive simultaneously to the different kind of triggers. For example, at the same time, they are sensitive to the pH, they are sensitive to the magnetic field, and they have a specific kind of ligands. Their stability is high, so we can rely on them. It's our perspectives for the future of the smart drug delivery. And also, the other important thing is about the biocompatibility. Most of them, as I told you, have some problems, toxicity, and they have lots of side effects for the normal cells. So we have to improve some biocompatible and biodegradable systems that have the lowest side effects for the normal cells. And also the other one is the integration with personalized medicine. It means that if you want to design a specific kind of nanocarriers, these nanocarriers should be just for you, not for another person, especially for your genes, for DNA data. And it will be very interesting that everyone with a specific disease have a specific treatment, but it needs a huge budget. And also real-time monitoring and diagnostic. It's very interesting if you, for example, inject a special kind of nanocarrier to the body. At the same time, it treats the disease, treat the cancer, and also you can monitor it. There's no need to imaging every time, every month, but you can track the nanoparticles, you can detect the problem, if there is any problem, and see it in a real time. And at last is our aim, always the aim of the scientist is translation of from the bench, from the lab to the clinic. And it's the last aim that we are trying to do it. As a conclusion and take-home message, these stimuli-responsive nanomaterials are great because they can target the specific sites, and we can control the delivery of drug to the specific site and at a specific time. And they have some responsiveness to the triggers, to the external and internal triggers. And also we can design them to increase the stability, we can decrease the toxicity, and the application of them was very vast, and they had different kind of applications in medicine and in every field, and it's very interesting. And at last, the challenges remained. We have to find a solution and overcome these challenges to make this development for the future and clinical translation. And here is the references, which are most important articles in this field of study. And thank you so much for your attention and for your time to listening to this presentation. And thank you so much, Dr. BETKAR, for organizing this webinar. And if there is any question, I will be glad to answer your questions.
Thank you, Mahtab Madam. Your research you have presented here with the best PPT. The stimulation, how it is useful in drug delivery, how the nanoparticles are responsive to ultrasonic, magnetic. If they stagnant in the body, they should be degradable. So all these factors, challenges of the scientist, all these things you have explained here, very useful one. And they are in treating the cancers from various organs of our body. Thank you. And we wish all the best for your future research. And I request all the participants, they can discuss with our speaker. Thank you.
Thank you so much, Dr. BETKAR. It was my pleasure to answer the audience questions. Thank you.
If anyone joined from your country, they can unmute and they can discuss. Sanan Shukla, sir, please.
Good evening, Dr. BETKAR, sir. Good evening, ma'am. Wonderful presentation you have made. And nanoparticle is really, you'll find it is one of the major thing what we can introduce in medicine and in the health aspect. So that is a wonderful presentation what you have made. These nanoparticles may be some chemical, physical, or maybe biological, or maybe the biological components we can use them. And what I understand that, there are, what you said, endogenous and exogenous are also there. But I will say, there are some primary nanoparticle and secondary nanoparticle. Primary nanoparticles, they will act itself as a, therapeutic, having their therapeutic value. They will be going to the targeted organ and they will work over there. And secondary, what I feel, they will stimulate the body, the body tissue or body particular cells or enzymes or hormones, to develop, and that is how it will work. So that is also nanoparticle. So I think, wonderfully well placed these aspects. No doubt in a health regard, we have to go for randomized controlled trial when we are introducing such a kind of drugs. So there are phases of the clinical trial as part 1, part 2, part 3. So randomized controlled trial, it is a golden standard by which we can identify what is the role of this nanoparticle, gold nanoparticles, silver nanoparticles, and n number are there. And people are still working and finding out and exploring, it is like a mine of, all gems. So we are searching for the gems. So that is also wonderful. You said one thing that these chemical substances should be biodegradable. So they will live their life and then they will get destroyed by themselves. So what I feel, the diseases like diabetes mellitus and other things, if suppose such a kind of nanoparticles, if we introduce and if they are controlling blood sugar level, that is wonderful. But to what length of time they will help? And if once they will get exhausted, by what we can say, degenerated in the body, again, we have to go for repetition of this nanoparticle doses. So that is what I feel, Sometimes you may find nanoparticles should, have a potential to maintain their, the therapeutic value for a little longer time. So they should, if we are taking biological component, it is very much possible they can multiply also, just like vaccines are also nanoparticles only. We are putting, the antigens directly. but no, there is no pathogenicity. So that is also one kind of nanoparticle. And I belong to the fraternity of homeopathy. I'm a homeopathic physician. So we are also using material, but the medicines, which we reduce them to the smallest extent. What we consider as in a physics, Avogadro number is there, but we process that Avogadro's number also. And I think that is also nanoparticle. In IIT Powai, we have done some experiment with this homeopathic medicines also. So please comment, about this biodegradable. And there are several questions arises like, the toxicity of this nanoparticle, then how they will reach to the target cells only, if there are malignant cells, how they will reach. Because, the way is blood circulation or lymphatic circulation, that is how they will reach. If they are taking a chemical or physical, they are not active by themselves like virus, bacterial components. So how they will reach to there? These are the several questions, comes in our mind, and whether they will really reach to that particular organ, because we know there are some, endocrine gland hormones. Hormones works on targeted organs only. Like, we are knowing that, the certain hormone, progesterone or estrogen, directly act only on ovaries. We are sure about this. It will never act on other organs. So these are the basic questions which arises as a researcher, right? So please comment on this. Otherwise, wonderful presentation, and it really helped me to arouse my own interest in the nanoparticles and this nanotechnology, what we should say. So with this, I will conclude, and please comment on this. Thank you very much.
You're welcome, and thank you for your attention and for your very complete explanation about this part. For entering the nanomaterials inside body, the injection is the way that mostly used. And after injection, there are many kind of ways to guide these nanomaterials to the specific location. And I told in my presentation that, for example, with increasing the temperature, some of the this kind of nanocarriers are sensitive to the temperature, and hypothermia is the way, for example, that these nanocarriers which are circulated are guiding to the specific location or accumulating, and this increasing the temperature will help them to release the drug. But we cannot say surely that all these nanocarriers will go to the specific location and release all the drug at the target site.
Yes, definitely. And I think we are having the immune system, strong immune system, when we are preparing some vaccines or something, we have to think about that immune system, because it tries to nullify and neutralize the foreign objects which are entering into the body. So that is also one thing. So you said injectable things are there. And no doubt, from the venous mode or arterial mode, we can reach to that organs. And how this nanoparticles will be having that affinity towards that particular organ. I think we can take a help of AI also nowadays. If we can implement some AI with the nanoparticle, I think it will become a super, technology in healthcare. So thank you very much, ma'am. Wonderful presentation.
Welcome. You're welcome, and thank you for your attention.
Thank you, Dr. Sir. Most terms that Mahtab Madam have explained, you understood. Just with your fruitful discussion, we come to conclusion that the drug delivery system with nanoparticles is important, and it should be specific one. It should not harm to other sides. So all these things are useful in the research. As you have said that AI should be implemented in this so that it will be more effective one. And our madam, Mahtab Madam also said about the challenges and in this field. So scientists and researchers are trying to solve the problems which are in the smart drug delivery. Thank you. And I am also thank you to all participants, those who are joined here. If anyone want to discuss, they can discuss. And if the participant joined from your side, from your country, I welcome all them, and they can come on the camera so that we have a screenshot.
Okay.
if possible. Thank you. And it's great to listen you. In future also you will be here as a speaker on another topic. And I request you, you can introduce your colleague, friends, those who are in the field of research. They can also deliver their speeches so that we can enrich in the knowledge every day. Thank you, Javed Singh ji. Thank you, Surya Vyas sir. Thank you, Priti Kumari. Thank you, Dr. Sanand Shukla sir. Thank you, Vyas sir. All those who are joined here from my country and from your country also. Thank you.
Thank you so much. Thank you, Dr. BETKAR, and thank you all other audiences. Have a nice day.
And those who are joined from your side, they are requested to fill the feedback link so that the certificates can be issued to them.
Okay.
I have read some names. I think they are from your side, so they can fill the feedback link, which is available in the chat box.
Okay.
And most important is that you have finished within 30 minutes. It's just 30 minutes is the protocol. But if the researcher want to have more time, then we allow it. But in next time, you can speak more time than 30 minutes. Thank you for your perfect presentation and best presentation today.
Thank you so much for your time. And I wish you have a nice day. Goodbye.
Goodbye. Goodbye. Thank you.
सर लिंक ओपन होत नाही है.
I think good and bye. We should say good and bye. Germany, they use the other term.
Okay. सर या लिंकला अजून 23 जण रिस्पॉन्स भरलेले आहेत, त्यामुळे लिंक ओपन होत नाही असं नाही. आता 23 जणांनी भरले बघा. तुमच्याच लॅपटॉप मध्ये किंवा मोबाईलला काही प्रॉब्लेम असेल.
ओके सर, नंतर